My name is Paige Coyles, and this is my description of protein synthesis translation. To begin, translation is initiated by the formation of an initiation complex, which is consisting of the smaller ribosomal subunit, the first amino acid, tRNA, and also the messenger RNA. The larger ribosomal subunit will then join the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites in which transfer RNA carrying amino acids can bind. One is called the peptidyl or the P site, and the other is called the acceptor or the A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transferred RNA that just entered into the A site. The ribosome will now advance a distance of one codon and the first transfer RNA is now released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred into the third amino acid. The ribosome continues along the messenger RNA as new amino acids are added to the polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid. This is what we call the stop codon. The ribosome will now dissociate it into the smaller and larger subunits. The messenger RNA and protein are now released.